Good afternoon, ladies and gentlemen. Welcome to attend the two lectures given by the two laureates of the 2014 Tang Prize in Biopharmaceutical Science, Professor James Allison and Professor Tatsuko Hongjo, for their discovery of CTLA-4 and PD-1 as immune inhibitory molecules that led to their applications in cancer immunotherapy. So what I'll do is I'll talk a little bit about their academic career and then a little bit about their research. And then we'll proceed to the lecture, first given by James. His topic is targeting immune checkpoints in cancer therapy, and then followed by Hongjo Sensei on academic innovation of cancer immunotherapy by PD-1 antibody. So you have 50 minutes, and there will be no questions. And after your talk, I will just give a very brief conclusion. So as you know, cancer has been the major cause of death in the past 30 years in many countries. Today, we see every year more than 50 million people have this disease, and that is a 70% increase compared to the number 4 years ago. And by the year 2030, we double. And every year we spend about 1.5 trillion US dollars on this disease. So today we'll hear some breakthrough in the field, particularly based on the discovery they made about 20 years ago. We know the immune system is very complex, but it's a very important, probably the most important defensive system we have in our body. And the process is highly regulated. And if it's too excessive, then we also have problem. So it has to have a balance between attacking the invaders and also maintaining the health of the systems. So two most prominent inhibitory molecules on T cells are discovered by these two scientists. They call CTLA-4 cytotoxic T lymphocyte antigen 4. And that is first identified on cytotoxic T lymphocytes. And then PD-1, program death 1, is first identified to be associated with the type of cell death process called program cell death. Now, their ligands are on dendritic cell or on cancer cells. They call B7-1, B7-2, or PD-L1 and PD-L2. So if the PD-1 on T cell interact with, for example, the ligand PD-L1 on cancer cell, then the immune system will be shut down, and the host, the immune system, cannot function as an anti-cancer defensive mechanism. So this is a very important discovery, and because of that discovery, that has led to the development of antibodies to interfere with the receptor and ligand interaction. So we'll hear beautiful stories from the two scientists in their contributions in the field of the fight against cancer. Now let me just briefly introduce, tell you the academic career of the two giants. One is James Allison. He received his BS in microbiology from University of Texas, Austin, and PhD in biological science from the same university. And then to my surprise, he went to Scripps as a postdoc. And after that, he became a faculty member at the University of Texas Cancer Center, and then he moved to Berkeley as a professor of molecular and cell biology, and then become head of that department. And then in 2004, he became chairman of the immunology program at the Memorial Sloan Kettering Cancer Center, and then he moved again to Texas. And now he's a chairman and professor in the Department of Immunology, University of Texas, MD Anderson Hospital, MD Anderson Cancer Center, and also director of the immunotherapy platform of the same institution. Now he received many honors and recognition. I just don't want to go through the list. Now in 1997, he was elected to the U.S. National Academy of Science, and in 2007 to the Institute of Medicine, and then 2011 he was awarded the Lifetime Achievement Award from American Association of Immunologists. And next month he's going to Canada to receive Canada Gardner International Award. Now, Professor Tatsuku Hongjo received his, I think, undergraduate and graduate degree from Kyoto University. And then he became a fellow of Carnegie Institution of Washington in the Department of Embryology, and then visiting fellow and associate in the Laboratory of Molecular Genetics at the NIH. And then he became assistant professor on the Faculty of Medicine, University of Tokyo, and then a professor there at the School of Medicine, at Osaka University. So he moved from University of Tokyo to Osaka University. And then he came back to Kyoto as a dean of professor and then dean of Faculty of Medicine at Kyoto University. In his career, he also served as a could be saying policy makers or policy association in many areas. He was director of Japan Society for the Promotion of Science. I think many of you are familiar with this. And an executive member of the Council for Science and Technology Policy Cabinet Office. And now he's a professor of immunology and genomic medicine at Kyoto University, Graduate School of Medicine. Again, he has a beautiful career and recognized by many honors and awards. In 1996, received the Imperial Prize and the Japan Academy Prize. In 2001, Foreign Associate of the U.S. National Academy of Sciences, and member of the German Academy of Science and Japan Academy. And he got the Robert Koch Prize and Order of Culture, Japan. And we are also very pleased that Mrs. Edison is here, and also Mrs. Hongjo. And so let's welcome the two scientists and Mrs. Edison and Hongjo. And as I mentioned, the first lecture will be given by Professor Edison. So let's welcome Professor Edison.
Thank you, Dr. Wong. Please applause to the speech by Laureate of 2014 Tang Prize in Biopharmaceutical Science, Dr. James P. Ellison.
Okay, thank you very much for the introduction, Dr. Wong, and I want to also thank the jury of the Tang Foundation for giving me the privilege of sharing this illustrious award with Dr. Hanjo for the work that we've done in the last several years. It's really an honor for me, and what I've learned about the Tang Foundation before and the visit, and during this visit, I'm really especially honored because it's a society that has a lot of other things besides this award that it does to better the future of humankind, and I'm really humbled and pledged to really be worthy of this prize. So as is mentioned, what I'd like to do is talk about some of the work that we did. Ultimately led to a new approach to cancer therapy. Before I start, I'd like to just point out that there are a lot of people who contribute to this work. The two most important, perhaps, are Max Krummel, who in my lab showed the negative function of N-acetyl-A4, and then Dana Leach, who in 1995 showed that that blockade of those signals could actually lead to tumor rejection. A lot of other people have contributed to mechanistic studies along the way, and then more recently, my colleague and wife, Dr. Padmini Sharma, has been collaborating with me on the topic of the last part of the talk, which we did together. And also the people at Medarex who took a chance on this new therapy in the late 1990s, Bristol Myers Squibb, who got into the game later and developed this commercially. All the doctors that were involved in developing it preclinically when they had to take a chance on giving this potentially very dangerous drug to patients. And finally, the patients, especially those in the early days who submitted themselves, again, to a particularly new drug that had never been in man before. So just to review quickly, from my perspective, I want to define what cancer is. And what that is a situation where cells become non-responsive to the normal constraints on their growth and proliferation and their location in cells. And this is caused primarily by genomic changes in the cells of two types. One are mutations that can be caused point mutations or translocations, both of which generate new things that have not existed in the cell before or change the function of molecules. For example, molecules like BRAF, which signal in the MAP kinase pathway and turn it on to drive a cell to proliferate. The other are epigenomic changes that can lead to derepression of genes and new things to be expressed. And the importance of these I'll get to in a moment. But cancer, really therapies for the past, since we've been treating cancer, have been limited to three modalities. Surgery to try to get all the tumor early on. We don't have to worry about what caused it. Radiation, which again, if you can get all the tumor, just kills the tumor cells by a beam of cells that cook the DNA of cells. Chemotherapy, which then usually just attacks rapidly dividing cells, or the more modern molecular targeting drugs, which basically target the specific gene products that are causing the cancer itself. So those have been the three pillars. And what I want to try to do to convince you today that immunotherapy has taken its place in the field as the fourth pillar of cancer therapy, something that many people have been working on for many decades. And why would we want to use the immune system to attack tumor cells? Well, for three reasons, basically, that the immune system offers that no other modality of cancer therapy can offer. The first is specificity. And I won't go into it, but I think everyone here knows that T cells are the warriors of the immune system, and they go out and they can recognize things on cells that oughtn't to be there, be they virus products or mutated products from tumors, mutated tumor genes or de-repressed genes that oughtn't to be there, and eliminate those cells by actually killing them. And so the immune system, and if you look at it that way, is really targeting the carcinogenesis process itself, because the T cells can detect the products of these mutations, whether or not they play a role in driving the proliferation of the tumor cell. So to put it in the sense that's used by cancer biologists today, the immune system can target the drivers as well as the passenger mutations in cells. The second thing that the immune system offer with T cells are memory. Once you've got those T cells that can recognize tumor changes in tumor cells, you've got them for the rest of your life, and you can reawaken them by some of the tricks that you'll hear about from both of us today. And finally, there's adaptability. The reason that cancer is so troublesome, particularly for chemotherapy and even the new genetically targeted drugs, that tumors can change, particularly when you have a large population of cells that are inherently genomically unstable. They can change, and so you target one gene and there's another mutation in it that makes it no longer responsible, or another mutation that activates a different pathway, so the tumor can get away from your drug. The immune system is designed to be adaptable. It's been calculated that the alpha-beta receptor, there are 10 to the 15th power different receptors that can exist. Of course, that's many billions of times more cells than you have in your body. But there's probably about 10 billion in everybody, just individual cells going around, each of which has a different one. Tremendous diversity that's generated by random mutations in the receptor genes to be able to protect you against anything that nature can throw at you, be it a virus, an infection, or in this case, cancer. So the immune system is at least, if not more, adaptable than a cancer cell if we just know how to regulate it. And so for decades, people have been trying to mobilize the immune system to attack cancer, but it really hasn't worked very well. We know from the work of Lloyd Old and others in the 60s that there are structures on tumor cells that can be recognized by T cells in mouse models. We know also, from the work of Lloyd Old and others, that there are tumor antigens in human tumor cells. There are T cells that can recognize those antigens in human tumors, and people have tried for decades to try to mobilize those cells by therapeutic vaccination, where you take something you know a T cell express and try to vaccinate with it by rapid, increased vaccination, but not in the way that you would do prophylactic to prevent somebody from getting smallpox, but actually to get the immune system rapidly mobilized to attack cancer cells. And that has been a dismal failure. A few anecdotal successes, but despite hundreds, literally hundreds of clinical trials, there have been no reproducible evidence of clinical benefit to patients by that sort of approach. So there's a lot of other varieties of that I won't go into, but it's been pretty much an abysmal failure. So what I want to do today is convince you that, at least in part, that's because we really didn't recognize how complex regulation of T cell responses actually is. So there was a mystery. I mean, people knew that T cells had antigen receptors. The structure of that was unknown until about 1982 or so, when we unraveled the protein structure of the T cell antigen receptor. But it became very rapidly clear, through the use of antibodies and other things, that this was not sufficient. You can't simply have recognition of something foreign by the antigen receptor and get it to go. By the late 80s, it had been shown by Rod Schwartz and Mark Jenkins and others that you need additional signals provided by things called B7 on antigen-presenting cells. Only these specialized cells can actually give the co-stimulatory signals. And again, in the late 80s, we showed that the receptor for those signals is a molecule called CD28. And a T cell has to get simultaneously the antigen-specific switch, or the ignition switch, if I can make an automobile analogy, and a push on the gas pedal from CD28 to get it to go. And yet there was another molecule that had actually been identified about 1988 by Pierre Goldstein, as you heard, called CTLA-4 because it was from cytotoxic T cells. But it became very clear, very rapidly clear, that that was a misnomer because it's in all T cells, but only after activation. There's none of it in resting T cells that gets turned on. So nobody really knew what it did until about 1992, 93, when Peter Lindsley at Bristol Myers Squibb proposed that it was actually, that it showed that it bound the same ligands as CD28, and proposed that it was another co-stimulatory molecule. And so that was the dominant idea for the early 90s until Jeff Bluestone and my lab independently conducted experiments that convinced us that that was wrong. The idea at the time was that T cells get activated through antigen receptor signaling and co-stimulation, and that they die by activation-induced cell death sometime down the road. But what both Jeff's lab and mine proposed, it was more complicated than that, that CTLA-4 can actually get in the way of co-stimulation and slow those T cells down, even stop them without killing them. And so this was quite controversial for a while, but was finally confirmed in about 1996 by Tang and others who made knockouts. And T cell mice that lacked CTLA-4 die of a lymphoproliferative disorder. So it's definitely one of the dominant downregulators of T cell responses. And so when we saw this result, first I'll just show you something I think is very interesting in how these work. The molecules are separated in time, right? There's no CTLA-4, the CD28's there all the time. CTLA-4 isn't there till later, after activation, but they also function very differently. Oh, sorry, if I could have the first movie, please. Here you see the CD28 molecule labeled with green fluorescent protein. That cell at the upper right comes into contact with an antigen-presenting cell. The antigen receptor signals, and you see the T cell, the CD28 move into the location of the T cell receptor where signaling is going on, and then provides the co-stimulatory signal that boosts that signaling pathway. On the other hand, CTLA-4, and I wish these would keep going, is at the distal side of the cell, and so vesicles containing it have to be drawn through the cytoplasm of the cell, where they come into proximity of the CD28 T cell receptor complex, and actually the bits of the CTLA-4 protein is then sprayed into the synapse and pushes CD28 out of the way and terminates co-stimulation. So as a biological process, it's fascinating, and we're still studying the basic biology, but for the purpose of today's lecture, I just want to talk about what we've done about that to think about cancer. Well, we noticed it was a negative regulator in about 1994 or so. I immediately started to think about its role in cancer, because it may have been the thing that was getting in the way of vaccination, therapeutic vaccination, because the CTLA-4 gene is hardwired. When you give a T cell receptor signal and a co-stimulatory signal, that turns on the CTLA-4 gene. So there's a period of time that you have for T cells to respond, proliferate, and go attack, and then CTLA-4 is going to dominate the response and shut it off, unless you do something about it. And so we had the idea that if you take CTLA-4 out of the picture by something as simple as a monoclonal antibody, that would take the brakes off, and you can control that by the half-life of the antibody and how many times you inject it, and let T cell receptor signaling and co-stimulation go unfettered for as long as you want, and keep responses going for much longer than you could otherwise. And the idea was that maybe you could get effective responses. And there were two things that were very important about this and compelling about it at the time. The first is you're treating the immune system, you're treating the T cell, you're not treating the cancer. It doesn't make any difference if you look at it that way, whether it's a KRAS tumor or an EGFR tumor or anything else. It's a tumor. The immune system doesn't care. It knows that there's mutations on it and can attack them. So it's a cancer drug, not a melanoma drug, not a c-KIT drug, not a RAS drug, whatever. And the second thing is you could use it as a monotherapy to keep an immune response going that's started by necrotic cell death, priming. Tumor cells, by the way, don't have the B7 molecule, and so they have to be phagocytized by professional antigen-presenting cells, their antigens redisplayed in the context of co-stimulation to start the response. And so the idea was you could use this as monotherapy perhaps, but even more exciting, you could use it to take the brakes off vaccines, you could use it to give permanence to the cell death and priming that's occurred by chemotherapy, radiation, or the targeted therapies. And so this was the hypothesis, and we tested it and published this in 1995, presented this. And this is actually a colorectal tumor growing on the backs of mice. It's an experimental tumor. You can see the size of it here. Check the tumor, it grows. You have to euthanize the mice after a month. You block the co-stimulatory molecule, the tumor grows even faster, showing that you have a nascent immune response that requires co-stimulation. But if you inject anti-CTLA-4, the tumor grows for a while and gets rejected. And this requires an immune response because if you deplete either CD4 or CD8 T cells, it doesn't work. So we saw this work. It was just astounding because out of all the things that are going on when a tumor is interacting with the host of an animal, you corrupt this one molecule and it turns growth and ultimate death into survival and permanent immunity. And so I'm not going to go through all the other animal experiments. We did a mechanistic studies in this time, but we did this against many different tumor types in mice, and virtually it worked all the time. Against many tumors, as a single drug. In other tumors were more resistant, we could use a vaccine, or we could use radiation, or we could use chemotherapy, or we could use other things to make it work. But we found virtually no tumor in mice that we couldn't treat with anti-CTLA-4 singular in combination. So about 1998 or so, we teamed up with Medarex, which had technology that had mice that had their immunoglobulin genes replaced by humans. And so we made a fully human antibody to CTLA-4. It was first called MDX-010, now it's called ipilimumab by the FDA. Subsequently, Bristol Myers Squibb got involved in this in about 2001 or so. But it's the fully human antibody. It's been given to over 50,000 people to date. There have been objective responses in many tumor types, including melanoma and many other types. And I'm going to concentrate, though, on melanoma today, but it has been, there have been responses in virtually every kind of cancer it's been tried against, just as we would have predicted from the mouse model. So this is one of my favorite slides because this is a woman I actually know. I didn't know her in 2001 when she was part of one of the early phase 1 trials, but I met her later. But anyway, her name is Sharon. She lived in Southern California. She had metastatic melanoma. You can see this tumor in her lung, this pleural effusion. She got a single dose of 3 mg per kg of anti-CTLA-4, again, in May of 2001. 6 months, and she'd failed everything else. 6 months after that, her tumors were completely gone. I met her in 2011, actually, by chance, I happened to be in LA, and her doctor, Tony Rebus, introduced it to me. That's her follow-up scan 10 years later, after a single treatment with anti-CTLA-4. And so this response lasted for a decade with no recurrence and no retreatment. And she's now 3 more years out. So she's about 14 years out now from having metastatic melanoma that was lethal, except for intervention with anti-CTLA-4. This will just show you different kinds of responses. This is another woman that has these cutaneous lesions on her legs. This is a patient of Jed Wolchok's, about 2004, from Memorial Sloan Kettering. You can see that, by the way, the standard therapy now is 4 injections, 3 mg per kilogram at 3-week intervals. So the therapy lasts for 3 months, and then it's over. It's not ever given again. So just so you know, it's not permanent treatment with this. So you can see this is after 2 doses of anti-CTLA-4, her tumors are almost gone. By 10 weeks, even before the finish of the treatment schedule, her tumors are completely gone. The only thing that's left are these melanin tattoos in her skin where the tumors used to be. And if you biopsy these, what Dr. Wolchok saw was nothing there but CD4 cells, CD8 cells. There were no tumor cells left anywhere in her body. So this is not, other patients responded more slowly, I won't get into that. But unlike other drugs also, patients with brain metastases respond. You can see this brain met here, which also resolved. So the response rate in brain cancer from melanoma metastases is the same as it is anywhere else. So there is no blood-brain barrier in this type of therapy. And it's not specific for melanoma, as you can see here, just to show you prostate cancer. This is a patient with hormone-refractory prostate cancer. You can see his prostate here, the bladder very shrunken there. He had treatment, all his cutaneous, all of his metastases disappeared. You can see the prostate is shrunk to normal size. So that was 14 months after treatment. Is still ongoing. There are two phase 3 trials that have been completed with anti-CTLA-4 in prostate cancer now and awaiting reporting, being analyzed. But I've heard that they look good too. So just to cut to the chase, Bristol Myers Squibb and Medarex planned a randomized, placebo-controlled registration trial, phase 3 trial of anti-CTLA-4 versus metastatic melanoma, and the results are shown here. This is an 800-patient trial. And so, this has got an extra line in it that I'm not going to go into. That was, anyway, anti-CTLA-4 plus a peptide. But what you can see here is the placebo control arm. You can see the patients rapidly die until the majority of them are gone by, are dead by 2 years. With anti-CTLA-4, there's no difference early on, but the median survival, the 50% survival, is about a 4-month increase. And so this is what you would expect to see from some of the FDA-approved drugs against cancer. You do a large trial, you see an increase in the median survival, and you say, oh, this worked. And so that would have been enough to get anti-CTLA-4 approved because no other drug of any type had ever given any survival benefit in this disease. But that's not the interesting thing. The interesting thing is that at 2 years, the survival is about 25%. And between 2 and 3 years, it drops to about 23% and stays there. So that's the tail on the curve. What that tells you is after 2.5 years, nobody dies from their melanoma. So this 4-month increase in the median just shrinks to unimportance in this particular trial compared, at least for these 20% of patients or so that live for the rest of the trial. And there was recently a 5,000 patient follow-up of that, and this is for 10 years. And the same thing holds for a decade. So 22% of patients with a single round of treatment with anti-CTLA-4 can look forward to a decade of tumor-free, well, not tumor-free, but a decade of survival without further treatment. Something that's unheard of, certainly in this disease. It's fully 20%. So that resulted in its approval by the Food and Drug Administration in 2011, and it's now the standard of care for treatment of metastatic melanoma. It says here, and I've mentioned this. So again, since it targets a T-cell molecule, it's being developed for multiple tumor types. Right now it's in trials in lung, bladder, and other kinds of tumor. But then I want to shift, and with apologies to Dr. Hanjo, talk about some combination stuff that's been going on lately. So CTLA-4, of course, is the first one of these molecules that was identified, but as you'll hear in detail from Dr. Hanjo, PD-1 and its ligands is another inhibitory pathway that's been in development and has had remarkable success. This was results that were presented in 2010 at ASCO. You can see this is metastatic renal cell cancer with shrinkage of this tumor, bone metastasis disappearing, the bone rehealing, something unheard of with other types of therapies that happens here with anti-CTLA-4. Here another tumor shrinking, and you can see as when the drug's given, there's infiltration of T cells. So PD-1 likes, anti-PD-1 antibodies like anti-CTLA-4 work in many tumor types. But just to make the point that I want to finish this part of the talk with, this is something presented at ASCO, the American Society of Clinical Immunology, by Suzanne Topalian in 2012. And this is monotherapy with the Bristol-Myers Squibb. There are 5 PD-1 antibodies in clinical development now and 1 PD-L1 antibody. But this just shows individual patients' tumors. And you can see the majority of the patients' tumors shrank here, some of them reaching objective at 50%. Some of these recurred. You can see at the red triangles, those are patients that had a new tumor appearing somewhere. Other ones. You can see there's nice responses of these, some of which last for quite a while after therapy. But PD-1, and you'll hear the details about this in a minute, but it was discovered at about the same time as CTLA-4 by Dr. Hanjo. Again, as was mentioned, it was known to first demonstrated to cause apoptotic cell death, programmed cell death, but it was developed also and shown to be another inhibitory point. But as the clinical success of anti-CTLA-4 became apparent, the PD-1 antibody began to move into preclinical studies in the early 2000s in mouse models. But just to point out a couple of things that are relevant to this discussion, like CTLA-4, it has 2 ligands on antigen-presenting cells and 1 that's on tumor cells. And this is very important because it gives it a whole other modality. But the thing that I want to focus on is that this doesn't have anything to do with CD28. It works apparently on inhibition of T cell receptor signaling. So the mechanism is completely distinct. As Dr. Hanjo has shown, it's completely distinct. There's no overlap at all in the way these things work. And so a couple of years ago, Mike Curran, a postdoc in my lab, was now an assistant professor at MD Anderson, reasoned that, well, the mechanisms are different. You ought to be able to put these things together and get increased efficacy. And sure enough, that's what you see when you start blocking these pathways together. In mouse models, it works better. But the really stunning thing was in 2013, last year, when this trial of anti-CTLA-4 plus PD-1 antibody was reported, and there were 2/3 of the patients now had responses in the sense of their tumor shrinkage. And you can see that from the spider plot at the top. Again, these are patients. It looks a lot different than the other one, right? So we're blocking 2. So 2/3 of the patients had tumor shrinkage. And of those, half of them reached the level of being objective responses, that is 50% shrinkage. And if you look down here in the waterfall plots, this is just showing patients that are looked at an index time. Each bar is a patient. So some of them grew, the majority of the tumors shrank. You can see half the patients that responded, their tumors reached the level of 80% of tumor shrinkage. And so this combination here shows just the power of this. A drug which there was no drug that gave a survival benefit. Now half the patients can be seen to obtain clinical benefit from a combination of these 2 drugs. Now, something that I didn't mention earlier when I was talking about the durability of anti-CTLA-4 was that the objective response rate, let me go back here, the objective response rate in terms of tumor shrinkage for anti-CTLA-4 in that trial, that phase 3 trial, was 8%, but the survival was 22%. The decades-long survival was 22%. So the objective survival rate, for reasons that are still not exactly clear, grossly underestimates the survival rate. And so this represents a 2-year follow-up of the experiment I just showed you with the combination of anti-CTLA-4 plus anti-PD-1 in metastatic melanoma, where 50% of the patients had objective responses. This is the same patient group. This is not randomized. This is not a large number of patients, so all that has to be taken into account. Nonetheless, at 2 years, and these are different dose rates, but at 2 years, at the optimal dose rate of 1 mg per kg of anti-PD-1 and 3 mg per kg of anti-CTLA-4, the response rate is almost 90%, the survival rate is almost 90%. So you have a 2-year survival rate of 90% now with these 2 drugs, when the survival rate previously was about 18% or so. So I think that if this holds true, if the CTLA-4 data can be extrapolated, I'm putting all these cautions in here because we don't know that yet, but these patients can look forward to maybe a decade or more of survival in a disease, again, where you were lucky to survive a year, 5 years ago, if you didn't receive these drugs. So I think that this sort of thing shows us that immunotherapy has definitely taken its rank with the other pillars, surgery, radiation, and chemotherapy, as a modality of treating cancer that can lead regularly in a large fraction of patients to durable responses, again, that last years to decades. And so where are we now? Well, just going off what I said, what we really need to do now is start looking at the cellular and molecular mechanisms that are involved in patients in the anti-tumor effects. We're doing a lot of that in mice, but we need to do that in patients as well. And maybe along the way, we can identify predictive, prognostic, or pharmacodynamic biomarkers that can help us evaluate what's going on in patients and something that's a little bit more valuable than just clinical responses. And finally, this will allow us, again, knowing the mechanisms can allow us to combine the best standard of care therapies with immune checkpoint agents. And also it may allow us to target new molecules to improve efficacy. And so now I'm just going to close by talking about some studies that I've been doing with Dr. Sharma. What Dr. Sharma realized many years ago, well, she didn't realize this, a lot of people knew it, but the conventional way of developing drugs is phase 1, where you just look for safety, phase 2, where you look for efficacy, phase 3, where you compare it to the standard of care and decide if it gets registered. Both the anti-PD-1 and the anti-CTLA-4 basically blow that whole model away by showing clinical responses in the phase 1. So right away, it's changed the paradigm. These drugs have changed the paradigm of clinical development. But still, that's the way that they're being developed most of the time. But if you do this, you can't really learn anything about mechanisms because the patients in these trials, you can get blood every now and then. Maybe you're lucky if you can get a biopsy, but typically you're not allowed to get tumor tissue from patients on these kinds of trials because they're not consented to do that. And so if you really want to do mechanistic studies, you have to design trials that will give you that as an endpoint. And that's what Dr. Sharma really pioneered by carrying out pre-surgical trials, if 1A or 2A or 2A or 1B, whatever you want to call it. But these are trials, and I'll show the details in a minute, where you administer the drugs to a patient before they go to surgery and then look in the tissue and see what happens. And this allows you to look then to get enough tissue, tumor tissue, to get enough lymphocytes to do functional studies and phenotypic studies and molecular studies, et cetera, to do biomarker analyses and understand mechanisms. And so, just to show the first trial that Dr. Sharma did of this type. This was in patients with localized bladder cancer. So these are patients that have bladder cancer. It's still in the bladder, so they're going to go to a procedure called cystectomy, where the tumors are removed. So the idea that she had was before surgery, the patients get 2 doses of anti-CTLA-4, and this is being done now with anti-PD-1, but in any event, then they go to surgery. We've got the entire tumor tissue, and then there are follow-ups where they get blood specimens and whatever. And so this just shows the idea of this. Again, instead of just getting a biopsy or bits of blood, you get the entire bladder from these patients. You get the entire tumor, except for the part that the pathologist has taken for a clinical determination. And that gives you enough tissue to get T cells, to do functional studies. T cells do flow cytometry. You can isolate fractions, do expression arrays, and see what's going on genetically. And this cystectomy, it just turns out that in males, the prostate is also removed. And it turned out that in Pam's trial, the men, half of them also had prostate cancer and didn't know it. And so from a single trial, there were 10 patients in the treated arm, 10 patients in the untreated arm, which is really all you need to get just an incredible amount of information. So she got bladder cancer, normal bladder, effects on the ureter, effects on normal prostate cancer, all in the one trial, 10 people. And so, again, without going into a lot, I'm just going to focus on one thing that both gives us a new pharmacodynamic marker of anti-CTLA-4 therapy, but also a new target. And so just to go through this, expression arrays revealed that the biggest change in gene expression were these T cell markers. You see virtually everything there is a T cell, and that's because there are virtually no T cells except for a small population of regulatory T cells in the tumor prior to administration of anti-CTLA-4. But after administration of anti-CTLA-4, 2 doses again, beginning 8 weeks before the therapy, there's this huge, this massive infiltration of the tumor. And the biggest change she saw was in the ICOS pathway. There's, of course, CD28, TCR, CTLA-4, all these other things. So this is just objective. This is asking the computer to tell you which is the biggest magnitude and where the biggest statistical significance is. It turned out it was in the ICOS pathway. And so what is ICOS? Well, oddly enough, this shows the phylogenetic tree. It's a very close homologue of CD28 and CTLA-4, although it binds to its own ligand called ICOS ligand. PD-1 is out here. Its other family members, B7X, B7H3, are out here. I'll just point out that of all these, the only positive molecules are ICOS and CD28. All the rest of these are negative molecules, which tells you something about how important it is to have tight control in turning an immune response off. There are more molecules involved in that by far than molecules that are involved in turning an immune response on. But anyway, what does ICOS do? What does ICOS do? Well, if you'd ask an immunologist, they'll say, well, these are follicular T helper cells. They're in the spleen and the lymph nodes. Dr. Hanjo's worked indirectly on these cells. They're required for class switching, which is something that he's defined over the past 2 decades. These cells provide the signals to the B cells that allow that whole process to go. It's also expressed on regulatory T cells, but it's never really been studied in cancer before. But anyway, this just shows proof at the cellular level that this happened on cells. So these are the bladder cancer, non-malignant tissues. If you look at the CD4 cells, about 13% of them have ICOS on the surface. Tumor tissues, same, about 16%. By the way, those of you who don't know these flow cytometry plots, each dot is a cell. This is represented by antibodies that have fluorescent dyes on them, different colors. And so this is just cells that express both CD4 and ICOS around here. But after the patients have been treated with CTLA-4 antibodies, you can see that goes up to 40%. So there's a big increase. You don't need statistics to say that that's a significant number. It's a 3-fold or so increase. And this also happens in blood. So this, she detected it in tumor, and then look for it in blood. And what you can see here on both CD4 T cells on the top and CD8 T cells on the bottom, in the blood, this also happens. Again, it goes up. In this case, about 10-fold or so here and 15-fold there in the blood as well. So this gives us then something that we can look at in the blood, because people that start looking at the blood in samples they've gotten from clinical trials don't know what to do with the data because they don't know what happened in the tumor. You cannot relate it to anything unless you do the tumor first, and you can say, is what happens in the tumor happen in the blood also? So can that tell us about cellular events in the tumor? And so this thing, the ICOS expression obviously can. And that gives rise to the issue of, well, okay, does this play a role? Is this a good thing for the tumors, for the immune response? In her trials, she couldn't really tell that because the patients had localized disease and were cured by the surgery. Although it turned out that 3 of the 10 patients at the time of surgery had no detectable tumor tissue, either by cytology or by genetics, and so either by histology or by genetics. So the patients were tumor-free, okay, but that's anecdotal. So at the time I was at Memorial Sloan Kettering, she shared this data with us, and Judd Walchuk, who had a large panel of patients, melanoma patients from the phase 3 trial that we had survival data on, we looked at those after Dr. Sharma gave us the information. And so we divided these patients into 2 groups, those of which had persistent increase of that population of cells, at least 2-fold above background, are those in which it didn't go up at all or only went up a little bit and came back down, had transient increase. And then if you look at survival of those patients, the patients in which it didn't go up or went up and came down, median survival was about 8 months. But the ones in which it went up and stayed up, it was closer to 20 months. So it looks then from the clinical data that this is certainly associated. Elevation of this population of cells is definitely associated with increased survival. But of course, one of the things about a clinical trial, an association of this sort, all you can do is generate the hypothesis that, well, maybe it has something to do with a therapeutic effect of NACTLA4. But you can't prove that hypothesis by looking at human tumor specimens. All you can do is say, well, that's a possibility. The other possibility has nothing at all to do with that, but the fact that these patients have all been heavily pretreated with cytotoxics, including many chemotherapies. I think the average chemotherapies that these patients got was about 4 or so different chemotherapies. It may just be that the immune systems of these patients have been so degraded by their prior treatment that they couldn't respond to anything, even NACTLA4. And so, again, you can't answer that question in clinical specimens. So that generates the hypothesis, though, that that pathway does play a role in the antitumor effect, which is a lot more interesting than just saying, well, the patient's tumors, the patient's immune system is too beat up to respond. So what we did then, Dr. Sharma took one of our animal models, the NACTLA4 in the mouse system with NACTLA4 plus a vaccine in this case, doesn't really matter, where normally you get about 90% to 100% cure rate of the animals in wild-type mice. But then the beauty of going back to a mouse model is we have genetically altered mice in which they lack either ICOS or its ligand. So they can't signal through that pathway. And so you can see right away the therapy is only about half as effective in mice that lack that signaling molecule. So that tells us, yes, the ICOS pathway is important in the mechanism of NACTLA4 enhancement of therapeutic effect. And then that, of course, leads to another hypothesis, and that hypothesis is that the ICOS ligand pathway might be targeted. So you know this molecule is important, so can you target it then? You block CTLA4, you take away the negative signals of CTLA4, you see the population of T cells that are expressing this new molecule. You know that that population of cells is important, so maybe you can give an agonist signal through the ICOS molecule now and see if it makes the NACTLA4 therapy work any better. And so there's a number of ways of doing this that we've been working on. We've done the first two, but because of time, I'm just going to show you the first one. And so what we did was to take tumor cells, in this case the B16 melanoma cell line, and transduced them to express the ICOS ligand gene. As you can see here, these tumor cells now express ICOS ligand. And the idea is that these will give the signal to that molecule when you're giving NACTLA4. And so mice get the wild type tumor, no ICOS ligand expression. And then after the tumors get to a certain size in the mice, they get the vaccine, which is either the wild type cells or the cells that are expressing the ICOS molecule, ICOS ligand molecule, and then anti-CTLA4 or not. And so then we see what happens when we treat the, when they're bearing tumors, the B16 melanoma. And these are the data, and it's complicated, but this just shows you that all the monotherapies do nothing. The wild-type vaccine plus anti-CTLA4 gives you a response rate of only about 30%. So the anti-CTLA4 treatment with only the endogenous ICOS ligand signaling, wherever that comes from, is not very effectual. But if you force the ICOS ligand signaling on ICOS by that vaccine that has the ligand on it, it goes up to better than 90%. And if you do that in mice that lack ICOS, it doesn't work. And so that shows that we are enhancing production now by targeting this new target, ICOS, in addition to blocking CTLA4. And so we're now working with a company that we founded to bring this to the clinic. We have some promising results so far. And I'd just like to say that none of this stuff I've talked about happens with PD-1. So this particular phenomenon is absolutely specific for anti-CTLA4 therapy. You don't see the appearance of these cells in mice that are treated with PD-1. So again, the mechanism, the cellular mechanism in patients in a therapeutic effect, like the mechanism of the molecule itself in inhibiting T-cell responses, is, as you might expect, completely different. And so we're trying to work now and figure that out. So this is now, actually this is out of date, but it shows the molecules that have been identified that are involved in affecting the functions of T cells during their activation. There's a lot of them. So the students in the audience, you know, there's plenty more work to do to work on these and really figure out the best combinations that can work either singly or together, which is where everything's going now. And so I'd just like to close with just sort of this challenge and this observation. Until recently, what we've been used to in cancer therapy is giving a drug, doing very large trials, hundreds of patients, and hoping to see an increase in median survival so you can say, oh, you've got X% of surviving 2 months, 4 months, whatever it is after therapy. There's nothing wrong with that. I mean, I'm sure that people would benefit, except we know now through the effects of anti-CTLA-4 and through Dr. Hanjo's work, PD-1, that we can get a fraction of patients that survive for quite a long time. And we know, again, from the combination of these two drugs, we can move that tail up, and this is just a curve not based on data. So our job now is not just to find drugs that will target these pathways, because we know it works now. We know that this is another form of therapy that can, for the first time, give us a fraction of patients with durable responses. And we know by adding them, we can increase it. So the challenge that we have now, and the promise for the first time in treating cancer, is to get that tail on the curve as close to 100% as we can and extend it to other cancer types besides just melanoma. And I think with the availability of these two pathways, we're well on our way. So I'll end with that and turn the podium over to Dr. Wong again. Thank you very much.
Thank you, Dr. Ellison, for the fantastic sharing. Ladies and gentlemen, please applause to the speech by Laureate of 2014 Tang Prize in Biopharmaceutical Science, Dr. Tazuku Honjo. Let's welcome Dr. Honjo.
Professor Wong, Tang Prize Foundation, it is my great honor and pleasure to be able to speak here as the first Tang Prize laureate. I'd like to start a little bit about my career. I have more time, partly because Jim kindly made a very nice introduction. So I can skip several of my slides, which I plan to use as an introduction. So I was trained as molecular biologist, unlike Jim Allison. So I did not work during my graduate school on immunology. I'm more interested how the gene expression is regulated. After graduation, after getting my thesis, I went to the United States and worked two great mentors, Don Brown in Carnegie Institution in Washington, where I learned eukaryotic transcription regulation. And during this period, he inspired me a lot. What is a fascinating story about immune diversity? Why we can respond thousands or even millions or even trillions antigens, yet our gene is very limited. Why that? This was a great question, and I got fascinated. But of course, it was very difficult to tackle this long-standing biological question. And actually, diversity is the essence of biology. We are here because our gene changed. Therefore, we are here. If our gene did not change, the same as the time when life evolved, all organisms disappeared because environment drastically changed. So I tried to tackle this problem, and I moved to Philides laboratory, and there I seriously worked why immunoglobulin gene change. And this is the beginning of the story why I got interested in immunology. And then I came back to Japan, 78, and continue to work on this question. And very fortunately, I found the enzyme called activation-induced cytidine deaminase, which regulate the immunoglobulin gene diversification, both somatic hypermutation that introduce point mutation and class switching that change type of immunoglobulin from IgM to IgG to IgA, and this is the basis why we have vaccination. Vaccination introduced enormous change in immunoglobulin gene. That was 2000. So then I like to go back to why we got into this PD-1, and I wish to tell you we are just lucky. We didn't plan. And we are very lucky, partly because technological development in biology was enormous. When I went to the United States, it was just time of the beginning of gene cloning, and immediately after, we learned how we can destroy the gene in animals, and then the era of gene sequencing. All these technology very efficiently allowed us to tackle the question which remained mystery for 100 years. Now, I'd like to go back to the story of PD-1, and I'd like to start from this slide. Simply, immune system is just like car. You have to start the car by putting the key. This is the ignition that is equivalent to the antigen recognition. And then when you want to move the car, you have to push the pedal of axle. And this is what Jim Allison said about co-stimulation, positive co-stimulation and CD28. And then definitely before we start, we need brake. Otherwise, the car will crash. So this is the target of our story, PD-1 and CTLA-4. And if the brake is too strong, the car cannot start. It's a parking brake. And this is the condition we think the tumor is growing. Our immune system is totally blocked, and therefore the tumor can overcome our strong surveillance by immune system. So this is the slides which Jim Allison already showed. Antigen-presenting cell express the antigen on MHC recognized T cell receptor, which is expressed on T cell, but they need positive and negative co-stimulation. And it's important, as Jim said, these two receptors, axle and brake, share the same ligand. That means they are competing. Now, that's not the whole story. As he said, we have ICOS as a positive stimulatory regulator and a PD-1 as a negative co-stimulatory regulator. So how we got into this story, in 1990 or 91, I forgot, one of my students, Ishida, who is a very bright young medical doctor, came to my lab, and he wanted to find out why our T cells are selected in thymus. And he wanted to know why over 90% of T cells are killed in our thymus during development. So he did this very simple experiment. This is the thymus-derived cell line, and stimulate it, and you can see very strong expression of the cDNA, which we named as programmed cell death 1. So this striking difference very much encouraged us. This must be something interesting, but we didn't know any function. by sequence analysis, we identified this is in this region, which is placed in the intracytoplasmic region, shares very interesting amino acid called tyrosine, which is often used as a target of phosphorylation and transduce signal. And all these molecules listed here are positive signal sending group, and the PD-1 share the tyrosine, but definitely different structure. It's a long insertion. So we still didn't know. But there were several very interesting observations. For example, this staining picture shows PD-1 expression in the area called germinal center, where the almost all antibody produced with somatic hypermutation and class switching. So if you look at this, this is an area called light zone where the antibody undergoes somatic hypermutation and selection. And this area overlaps with T cell, which express CD3, and B cell, which express CD20. So this unique expression profile and strong induction of PD-1, as you saw before, convinced me we have to do something to find out this function. There must be something new. So the most straightforward experiment is to destroy the gene for PD-1 and see what happens. And Dr. Ishida and Agata and also Nishimura spent years to knock out a gene and study the animal. And they were very much disappointed because nothing happened for the first 3 months. And then waited longer, and also they made mice genetically pure. And finally, they got very striking observation. Excuse me. So this is the black mice, and they show nephritis and arthritis. And white mice, you can see the size of the heart is very much enlarged, and they die. NOD mice, which is well known as a model mouse for diabetes, they have very severe insulitis. And MRL mice, which has a myocarditis and also become very bad. So these results, and together with, I'd like to go into a little bit of these two events in what happened in white mice. This is the section of the heart, and you can see very strong enlargement of the heart, and the muscle fiber are very much disturbed. And what happened is actually, this is the antibody deposition on the heart tissue. This is the antibody-produced mice without PD-1 gene. These antibodies are against their own heart and accumulate as you detect by immunostaining. This green fluorescence indicates. And the complement, which comes with antibody, also accumulate. And this will destroy the tissue. And we identified this is the specific antigen recognized by self-antibody, and that is actually troponin I. And even our student, Okazaki, made antibody against troponin I, injected into mice, and he could recapitulate by antibody administration. You can make mice sick with enlarged heart. So I think many patients who suffer from dilated cardiomyopathy is type of autoimmune disease. I try to convince the cardiologist, instead of replacing a heart, you better treat them as an autoimmune disease, but they never listen to me. Okay, the other model is I just showed NOD mice, and NOD already becomes sick But when we cross with PD-1, as you can see, in NOD mice, male and female are certainly different. So female and male profile, but basically the same. In the absence of PD-1, they become sick earlier and more severely. This is the severity of the animal, and this minus means both allele of PD-1 is deleted. You can see very much severe phenotype observed. And most striking, I was very much interested this PD ligand expression profile along the islet, and this red T cell, helper T cell, blue CD8 is the killer T cell. So all these are blocked by PD-L1 expressing barrier of islet cell. But when you knock out PD-1, there's no barrier and lots of CD4 and CD8 invading into the islet and causing very severe insulitis. We then found, so this indicates that our immune system is very much accelerated without a break. Now we can test this by giving them virus, the blue labeled adenovirus in the wild type. After a week, you can clearly see lots of virus in their lung. And even 30 days later, still you see a virus. But if you don't have PD-1, they clear the virus very rapidly, indicating our immune system is very much accelerated in the absence of PD-1. So then we need to understand more seriously the mechanism we need. What is the molecule that stimulates PD-1? And that is the ligand which binds to this receptor. And fortunately, 2000, we in collaboration with Genetic Institute and Harvard Medical School, Dana-Harvard, we were able to identify two ligands, PD-L1 and PD-L2. And using this ligand, we can study the mechanism of PD-1 signaling. So the first experiment, this shows CD3B7, this is the molecule you already heard Jim Allison, just gives positive stimulation over CD3, that is T-cell receptor stimulation. And in case of PD ligand L1, actually, this is a negative stimulation you are seeing as cell proliferation by thymidine incorporation. And clearly, this co-stimulation with against T cell and PD-1 together, you get reduction of cell proliferation. So Tako Okazaki tried to mimic this using interesting strategy outside FC gamma receptor, inside PD-1, and cross-link with immunoglobulin recognizing IgG using a B cell as a model. And if they cross-link, they send the signal and suppress B cell receptor signaling. This is the same with T cell receptor. Instead of immunoglobulin, they use T cell receptor. As a control, immunoglobulin without FC portion, which blocks the binding to FC gamma receptor outside of the PD-1 molecule as artificial fusion. So by this method, he can show immediately after 8 seconds, he can see the phosphorylation of tyrosine inside the PD-1 molecule. And if you change tyrosine to phenylalanine 1, 2, both you see clear reduction of phosphorylation. So this phosphorylation by cross-linkage is the mechanism. And then he found the protein which is recruited by phosphorylated co-stimulation phosphorylated PD-1 is SHUP2 phosphatase and not SHUP1 or SHIP. And again, mutation kill this SHUP2 recruitment. So taking all these together, the mechanism of PD-1 is very interesting. Again, as Jim said, this is totally different from CTLA-4. By stimulation of PD-1, they phosphorylate, recruit phosphatase that remove the activation signal through antigen recognition. So this is a very striking difference from between PD-1 and CTLA-4. As I understand, Jim may correct me if I'm wrong, the expression profile are not so much different, expressed on both T-cell activated and some FOXP3 positive T-cell, except B-cell expression PD-1, I never heard CTLA-4 expressed on the PD-1. The most striking difference is the signaling mechanism is, as I showed, it's using phosphatase. And CTLA-4, there is some controversial literature, so clearly ligand competition between activation and negative is difference. And some reports says SHUP2 is also recruited, and some reports says IP3 kinase is activated. So I'm a bit confused on this portion, but the clear difference is this phosphatase-mediated gradual downmodulation of activation versus competition between on and off. It's one is more clear on and off, the other is gradual rheostatic regulation. The phenotype is also very striking. As I mentioned, the PD-1 deletion causes rather mild and long-term autoimmunity, but as Jim said, CTLA-4 knockout is very severe. It's a very critical difference. Now, after these studies, which we completed around 2000, we had a very naive idea. If we can upregulate immune system, we may be able to even attack tumor by simply blocking break signal. So we did experiment sometime early 2000, and here the expression profile of PD ligand on different tumor cells of mice. So look at this. This shows toward the right side, this peak shows very strong PD ligand expression. And we used these mice, tumor cell, inject into bulb C and measure the growth of tumor versus day after. And this is the control and tumors growing happily. But if we inject PD ligand 1 antibody, the growth is very much suppressed. Similarly, same tumor injected bulb C, wild type versus PD-1 deficient, same line, you can clearly see tumor growth is completely blocked. Different tumors which originally didn't express ligand, but we forced to express PD ligand, and then similar thing happens by giving anti-PD ligand blockade antibody, you see reduction of the tumor growth. and survival curve clearly showed the clear difference. So this is a very impressive and surprising observation for us, and we published this result 2002. And the experiments was done by Yoshiko Iwai, who further pursued this and showing the melanoma metastasis. And this is the size of liver which was isolated after injection of melanoma into the blood, and the PD-1 deficient animal clearly blocks the metastasis of melanoma circulating from the blood. We then thought whether this is only true for mice or this could be true in human. So we collaborated the gynecology department, Hamanishi, at that time, I believe graduate students, now the assistant professor in Kyoto University, went back to the patient who had operation of ovarian cancer. So you go back to the patient sample and stain whether each patient tumor expressed the ligand high or low, and studied how long the patient survived after operation. And we found very clear difference, statistical difference. And similar study was actually published by a number of other groups: renal cancer, esophageal cancer, gastric cancer, urothelial cancer, and pancreatic cancer, and melanoma. All shows the tumor which expressed PD ligand somehow showed worse prognosis compared to those who didn't. So, putting all these together, we are pretty much convinced there must be some clear regulation of tumor through killer T cells. Killer T cells recognize tumor antigen and attack, but sometimes tumor express ligand against the PD-1 and block the activation signal within the T cell, and that causes so-called tolerance, means parking brake onto the T cell. If we introduce antibody to block this interaction, we may be able to rejuvenate this T cell and attack again. So we wanted to apply this strategy to patients, and we obtained patent and tried to convince pharmaceutical industry, and it was not that easy because at that time, most of the cancer specialists considered immunotherapy is very shaky. So company won't invest huge amount of money. But we managed to persuade, and Ono collaborated with Medalex, the same company. Jim collaborated. Now it's bought by Bristol Myers, and they successfully generated and get approval from FDA as investigation new drug, and they immediately started clinical trial. They started 2006, and in Japan, Ono also started the clinical trial 2009, and the outcome partly covered by Jim, and it's very striking. And the most striking first paper appeared in New England Journal of Medicine 2012 by the Johns Hopkins group. And this clearly showed the over 296 close 300 patients having the non-small cell lung cancer, melanoma, renal cancer, and all of them showed reasonable response, 18 to 28%, as Jim said. But this is rather surprising for the cancer specialist. One drug covers many different types of tumors and with close to 30%. And most striking, again, Jim already mentioned, is durable response, lasts long time, over 3 years, is really surprising. So I'd like to introduce our own study in clinic in collaboration with gynecology, again, Junzo Hamanishi, and many others. So we try to extend our early observation with ovarian tumor and made a small clinical trial, phase 2, and had 2 cohorts with 1 milligram per kilo and 3 milligram per kilo, 10 patients each. And we recruited patients if they are not having any autoimmune disease, and if they have kind of the very bad stage after platinum treatment, no clear response, and has a reasonable size of tumor even after surgical treatment. And so the system is we inject anti-PD-1, which is the name after BMS, nivolumab, and every 2 weeks, and follow up the patient every after. So the clinical study finished and now at the stage of making a final report. There were some serious adverse events, fortunately no deaths. And the outcome is 70% is good response. If you combine those which did not increase the size of tumor, stable disease, that it goes to 44%. So this level of the response in ovarian tumor is very surprising. And I'd like to show you some of the individual case. So this is the case of the 60-year-old lady, stage 1C, which I don't know exactly, but the progressive disease after lots of treatment. And the CT scan shows huge tumor. And after 4 months, this is completely disappeared. I'd like to stress, this is a clear cell ovarian tumor, which all the gynecologists said it's impossible to be cured. And again, oops, this is the marker is very clear reduction. Sorry. So another case, this is serous ovarian tumor, and again, she had the clear sign of tumor which disappeared after 4 months, and the marker goes down very quickly and stays low again more than a year. So putting all these together, this is the picture like Jim Morrison showed. Among 20, actually 18, I think, is the complete response and the partial response patients, and some stable disease level, and some clearly no sign of response. Again, this is the most important sign of this immunotherapy, durable, and so this is not so old as compared to CTLA-4, only the more than year and a half. Still, they are very happy. And when the TV team from Taiwan came and they wanted to shoot the patient treated by this therapy, the lady who received this treatment, who lived in Osaka, and the doctor was afraid to call her, but once he called, she was very happy to come and got an interview. So that means she is so good and so much pleased to get through this therapy. So I think I can skip this because Jim already showed combination therapy with CTLA-4 shows more dramatic response, durability, and also percentage of response is impressive. So now, this therapy targeting PD-1 or its ligand is a target of many drug companies. Originally, it started Bristol Myers and Ono, but now Merck and others, even Roche and AstraZeneca, all major companies jumped in. Well, I think there is a good reason, because if this treatment are targeting all kinds of tumors shown here, this is the list of clinical trial clearly approaching to the completion, and hematopoietic tumors are just started. All kinds of Hodgkin's, and those are responding very well, what I heard. And colon cancer, hepatoma, pancreatic, gastric, mammary tumor, all these are underway, so sooner or later they will be approved. So under these conditions, not a single company can offer or provide enough product to the market, and that's why many major pharmaceutical industry jumped in and will provide the medicine to the market. So this is the just history, what we have went through. 1992, we found PD-1 and found its mechanism and identified the ligand, and in animal model, we found cancer can be treated. And we also showed this is effective for prevention of infection, and I'm trying to persuade people to use this for HIV treatment. And company made humanized PD-1 antibody 2006, and clinical trial lasted several years, and finally this year, this PD-1 antibody was approved by PMDA in Japan in July, and FDA in September this month. So this shows, as I said, it takes long time, and we need basic research investment with a long-term vision. I like to summarize the advantage of PD-1 therapy is, first, it less adverse side effects. And probably this is because of rheostatic regulation. It's not just strong break, it's gradual break. So that will be good for our own body. And second is effective for a wide range of tumors, and also it's sustained for long term. And these 2 points are probably the advantage of our immune system, because we have enormous diversity, as Jim said. The power is 10 to 15 or it could be 16, and that can recognize even if tumor change themselves. This is a major problem for chemotherapy. Chemotherapy works for first 1 month or maybe 3 months, but tumor change, mutate, but T cell can have enough repertoire to attack newly modified tumor cells. Well, finally, this is the future possible combination with anti-cancer treatment. This will certainly enhance the efficacy of this treatment. So we offers one of the best example of scientific translation from bench to bedside, and a powerful demonstration of the extreme importance of basic research for progress in medicine. And thirdly, long-term support. I'm just saying this for many cases. This is because everywhere in the world, somehow politicians are looking for, except grow, very short-term outcome. And this is very harmful for science. And that's what the reason why I'm stressing this. So the final slide is what is the future of this treatment? The first, although it's mild, the side effects, autoimmune symptoms, inevitably develop and must be carefully watched. I'm just warning this because most of the cancer specialists don't understand immunology, so they tend to see the effect. And the autoimmune diseases develop in a various way. So you have to be very careful to treat the patient with this immunotherapy. Second, it's very important, again, Jim mentioned, why there are non-responders. And this is a very important and a critical question we have to understand. And thirdly, important if we can identify markers. And this will save lots of money and lots of time for patients and lots of time for clinicians. So it's ideal we can tell whether this particular patient will respond to this treatment or not. So I'd like to thank my collaborators. Dr. Iwai, who is now the professor in Kyushu, initiated animal experiments for the tumor treatment. Nagahiro Minato, professor and dean of Kyoto University Medical School, who unfortunately have to leave this morning, but he originally, together with Yoshiko Iwai, started together with us the animal experiments to treat the tumor. And Nishimura and all those, oh, I have to show Ishida is the one who insisted he liked to clone the PD-1, and he succeeded. And I mentioned Okazaki, who identified molecular mechanism on PD-1, and this is the, I cannot read all the names, but the people in the gynecology group and medical oncology group and all those hospital personnel are critical to carry out the clinical trial for ovarian tumor. I'd like to close and thank you for your attention.
I hope you enjoyed the two lectures, very exciting and inspiring. The lectures are so easy to follow, so I don't think I need to further elaborate. But I think the opportunity should go beyond cancer, as you mentioned, that could include autoimmune disease and infectious disease. One thing I want to comment here is that from the story, you can see that the discoveries were made in the early 90s, around 92. And then the proof of research proof of principle was about 5 or 6 years later. And then the development of the antibody for the treatment of the disease is even later. So 20 years from discovery. And the first antibody against CTLA-4 for melanoma was approved in 2011. And the antibody against PD-1 for melanoma was approved this year, first in Japan in July, and then September 4th this month by the US FDA. So that tells us that it takes a long time for discovery research to be realized for the benefit of mankind. And I hope, I don't know if there's any politicians here, funding agents here. I think it's a good lesson for us. So congratulations to both of you for the award and the remarkable work, and also to everybody for your participation. Thank you.
Thank you, Dr. Wong, Dr. Hongzhou, and Dr. Ellison. 谢谢两位得奖人以及主持人精彩的分享。第一届唐奖得奖人演讲生技医药场到此圆满结束。